yeah, I'll start with this video of Drosophila that you've probably seen way too many times already in your life. But yeah, this is, if you haven't seen it, it's like the Drosophila embryo invagination. So you see like these large scale deformations and flows. I want to point out two things that are maybe less well known. Okay. The first thing is that if you take this embryo and you just look at the wild type, you see this initial deformation here that triggers this invagination. And if you zoom in there, you can see some local enrichment of actomyosin. It's a bit hard to see, but you get this deformation. At the same time, you get this localization of actomyosin. And what they did is they looked at the mutant where this process doesn't happen. And if you just look at the mutant, it just doesn't deform and you don't see any local enrichment. But then if they mechanically perturb it, mechanically indent it, they recover this local enrichment of actomyosin. So if they perturb it enough, they get something that locally looks like the wild type as well. So just by mechanically indenting it, you can get this enrichment of actomyosin locally. And then the more recent work is this morphogenetic wave that was proposed to explain this, where initially cells are deforming, so there's some contractility that leads to the compression of the neighboring cells, which then leads to the activation of actomyosin and deadhesion from the substrate, creating this feedback loop that gives you this wave that goes here from left to right and creates this invagination in the center here.
Good summer. Good summer.
Thank you. I was very afraid of what you would, which mistakes you would find.
I just want to say that
yes,
the experiment at the top right,
yeah,
just take it with a grain of salt.
Okay, that's why no one cites them, or
yeah, exactly.
Okay, I was wondering about that. Seems very ignored in the literature. Okay, okay. Good to know. Sorry? This is the actomyosin. Look.
Like, what's the actual probe? Like, when you say actomyosin, what am I looking at?
Like, how is this?
Is that myosin light chain? What are you labeling with green?
I don't know.
Well, that's part of the grain of salt. Yeah.
Okay.
I don't know.
What did you use? No, we use in our case it's pretty clear. It's a myosin regulatory light chain fused with GFP,
okay,
which is at endogenous levels, etc. Top, this is antibody staining.
Okay. Okay, what I want to draw from that is that there is some correlation or like interaction between mechanochemical feedback and deformation. And if you trigger one, you can get the other and so on. So they talk with each other and somehow. I hope that is uncontroversial despite the questions about the first paper. Okay, so we want to, inspired by that, create a simple model where we start with a thin cell sheet. We impose periodic boundary conditions. So you can imagine it's wrapped up in this cylinder, similar to the Drosophila embryo, or otherwise just as a mathematical tool if you make the curvature very large. And
curvature very small.
Yes. Radius very large. And then we just take a one-dimensional cut, so cut through the cylinder and look at this one-dimensional ring that we get, which we assume to have three properties. The first one is that it deforms viscoelastically. So if you apply if you apply the white external forces similar to the white arrows here, it just viscoelastically deforms according to the external forces. And then we put in some activity, which is that locally you can imagine there are these microscopic contractile force dipoles that could be, for example, actomyosin. And the effect of them is that if they are distributed symmetrically in this ring, then nothing happens and the configuration is stable. If the configuration is asymmetric, then you get this deformation because at the top here you have contractile forces that locally deform you, and at the bottom you have nothing that acts against it, so you deform into, or you bend in this direction. And the other property is that this concentration is stress dependent. So if you start with some initial conditions and you pull on them, then either you increase or you increase the concentration if you apply forces, and then that's called a catch point, for example, or you decrease the concentration when you apply forces. And it's called slip points. That's some modeling choice, which of the two you would like to model, and adjusted sign difference, so we can include both. And
I think that the evidence for the myosin in general, that
Yeah.
is a strain-dependent stabilization.
yes, exactly. That's kind of the idea, that as you pull on it,
Yes.
they become more stable.
The duty ratio is changing. So
Exactly. So we have these three things, and then to model exactly that as a first part, we can look at the stress-dependent concentration. So in the simplest case, we have the concentration N that dynamically over time changes, increases, and dissociates with some constant alpha, and then for long times, in this case, you just evolve to this constant value. And to put in a stress dependency, you can add the stress-dependent dissociation rate here with, you apply some external stress sigma here, and there is some threshold value, which means that if you are above the threshold value, the dissociation rate in this case is very small. So you go to the same value as before, and the stress has no effect. Whereas if in the opposite case, the dissociation is very large, and you completely dissociate.
why did you choose stress as opposed to strain-dependent stabilization?
It will be the same in the end. So in the I'll show you in the next slide, but it will be basically local stretching, because it's one-dimensional. It's the same. So as for a general stress tensor, if you only have concentration, then the natural choice to put here is that because all of these local force dipoles are pulling on each other, they are exerting forces on each other. So you can just model them as some spring that is, the strength of the spring is proportional to the local concentration. And if you plug that in here, you get this two possible solutions. So if the initial concentration is very small, it fully dissociates and reduces to zero. But if the initial concentration is sufficiently large, then you have sufficient stresses in the system that you actually increase your local concentration because of that, and you go to the other fixed point. So you have two stable fixed points, or two stable solutions in this case, as opposed to just one before. And this threshold here that tells you how strongly do you have to pull to make any effect just sets the fixed points that you have. So for very large threshold, you only have the solution near zero, so they will always all decay to zero. For very small thresholds, the only possible solution is one, so you're kind of back to the constant dissociation, and you just go, everything always goes to the constant solution independent of initial conditions. And then for intermediate values, you have these three fixed points, and depending on the initial condition, you either run into one or the other. And that's kind of the interesting case, of course. And to couple that to this elastic ring, we assume that the geometry is relatively thin, so some height h, and then a quasi one-dimensional coordinate system along the length of the ring. And the equation describing the ring is given by this. So the first two terms are just Laplace equations. So the local curvature is just given by the external, or the pressure difference, and the surface tension. And then there is some elasticity entering here. And the way it enters is that, as you mentioned, just through the local strain. So if you take an elastic ring, you bend it slightly, it will be stretched below and compressed above, and this will try to push you back into the straight configuration. And you can write down the resulting moment that drives you back to the flat configuration. And it's just some constant that determines the stiffness times the curvature. So the stiffer it is, the more force you need to exert to deform it to the same state. The viscous bending moment, you can do the same thing. you just add some time components. So no, what matters is not the curvature, but the rate of curvature change. And otherwise, it's exactly the same. And then say it again. This parameter just determines how quickly do you evolve to the final configuration. So that's just a ring that deforms viscoelastically. And then the activity comes in as this active moment. So we take the stress that we had before, which is proportional to the concentration. So you take these force dipoles everywhere, you integrate them over the width similar as before, and then you get the equivalent active moment. And yeah.
Just briefly, I'm trying. Are you adding these two components in parallel or in series? The way that you've written that. In other words, is it viscous on the longest time scales, or is it, which would be if these two components are added in series, or is it elastic on the longest time scales when they're in parallel?
It's elastic. So as you, yeah.
Okay, so you're doing like a Kelvin Boyd model.
Yeah,
yeah.
So it just sets the time scale that you need to evolve to the final state that's set by your elasticity. So yeah, and then the active moment does what you would expect, what we've seen in the video before. If there's an asymmetry in concentration, there is contraction on one side, but not on the other side. So in this case, the active moment would drive to flatten the ring. So to summarize, that's the model, the concentration equation coupled to this viscoelastic active ring. And then this plus minus here is this choice that I mentioned before between should applying stresses increase or decrease the concentration. And I'll show the effect of that in a second. So to see or to get an idea of what happens here, we can for now just treat this parameter, this curvature, as a constant. Yeah.
Maybe quick clarification.
And
can you go back to your first slide? So what assumption do you make for the profile of n across the thickness?
None. I mean, you have some initial condition, which is constant, but then you say that it locally evolves according to this equation, where locally you just have the active stress, the elastic stress, everywhere determining which solution are available.
3D or 2D at least.
Yes, exactly. You solve this in effectively 2D. Yeah. Okay, so to understand what happens, we can for now just look at a ring with this concentration equation, maybe to answer your question as well, and just treat the curvature as a parameter. So we just take the ring and we deform it by hand, and we don't let the ring deform on its own. So if we start with some initial condition, then we can compute the active moment and the elastic moment over time. And initially, we just randomly choose this concentration. So there are some contractile forces kind of bending you downwards. And then as you deform this ring by hand, you record what happens to the active moment, to the elastic moment over time. And if we look at 3 snapshots, then as the ring is deformed to the flat state, you see that the concentration increases on the other side. So this active moment completely disappears. And as you're in the straight state, there is no moment either way. And then if you deform it further and deform it the other way, the concentration inequality appears again. So now it's contracting in the opposite direction. And what that means is that as you deform it, the active moment is helping you deform it, because it's here it's contracting in the same direction that you are contracting it while you're deforming it by hand. And you see the same in the equation. If you add up the active moment and the elastic moment, you get something that's smaller than it would be if you just had the elastic moment. So what that means is that the ring softens due to the activity compared to the passive one. And then for the negative feedback, which is, the plus sign here, the positive feedback was the minus sign here. And this would correspond to the catch bond microscopic behavior. We start with the same initial condition. We deform it by hand again. And as you might have expected, you see the opposite happening, that now the concentration is acting, the active moment is acting against the deformation that you perform by hand. So compared to the passive one, the ring stiffens. So that's treating the curvature as a parameter. And now we can, hopefully having a better understanding of what's going on, go back to the full picture and look at the three coupled equations where the curvature is a dynamical parameter as well. And to do that, we take the elastic ring, we deform it in this with this pressure. So we compress it at the top and the bottom, we stretch it at the left and the right. And then we increase the magnitude of this deformation. And we can also, as a second parameter, change the threshold parameter. It's possible to write down some analytical prediction for the onset of instability. So how far do you have to deform the ring for something to happen? And then these color dots here are the simulation results. And what that looks like is that if you are in the region where there's no feedback, you deform the ring, and compared to the passive one, it looks exactly the same because the feedback is not triggered. Or if you go into the region of instability, then for positive feedback, you see that now the deformation is enhanced compared to the passive ring. So you deform it, and at some point you trigger the feedback, and then the feedback is locally softening the ring, so it keeps deforming on its own, and you get this runaway feedback mechanism where local deformation results in local activation and then more and more deformation up until some point where it stops on its own. And to look at the opposite case of negative feedback, okay. As you might expect, the opposite happens, and because the ring is locally stiffening, now the deformation is less than it would be in the passive case. And something fun happens if you turn off the external force. So now we do the same as before. We turn on the external perturbation, deformation. We look, and then at some point we turn it off. So we remove the external forces. So this is the same thing we saw before. The active ring deforms more, and then the passive ring, as we turn off the external forces, goes back to its initial state, as you would expect, but the active ring stays permanently deformed. And something similar happens for the negative feedback, just that because of the way the feedback operates, it is now stretched in the direction where you initially compressed it. So somehow by having this feedback, you can turn a local compression into a local extension of the ring. So that's the result of the model, theoretical model. We wanted to see if there is any, if this is actually observable in experiments. So Ray at the medical school here was studying the trachea-esophagus separation, which is the developmental process that happens where the trachea and the esophagus separate over time. So you can see in a chick here, you go from having one connected tube to this budding off. So the splitting of the tube and then these two tubes growing independently, and one will develop later on into the stomach, and the other one will develop into the lungs. So in the first step, Ray, to study this process, took a two-dimensional slice through the tube at the position where it is not yet separated, and then studied this two-dimensional slice over time. So kind of in this two-dimensional setting here, the growth is being replaced by the time evolution. And what you see is that this epithelial ring that is initially containing one lumen over time separates into two lumen. We can study the curvature evolution over time with the same caveats of having noise that Suraj was talking about before. But it works kind of well. And if you want to read more about it, the work is published to see how, as you perturb the mesenchymal cell surrounding it, affects the curvature evolution and prevents the splitting from happening or does not. Yeah.
In this example, mesenchymal tissue around is pushing.
Yes, that's.
Do you know if it's pushing everywhere or if it's pushing?
It's pushing primarily
there
near the region of the neck where the transition happens. And well, the whole thing is about as you perturb the external cells and you prevent this pushing from happening, the transition does not happen anymore. So you can kind of just mechanically cut off the cells and you do not see this transition happening. And then you can locally push by hand the epithelium together. And then if you push it sufficiently, it goes through the transition and keeps evolving as you would expect. So yeah, that's about the effect of the external forces. And then to connect to the model that I was talking about before, Ray cut this epithelium ring at some point and then deformed it by hand, measuring the concentration of actomyosin before and after the deformation. And then you get this kind of relationship where as you decrease the concentration, the active moment that you compute from the measured actomyosin reduces as well. And comparing that to the prediction of the model, you see that it has the same behavior roughly as the negative feedback would predict. So there's some evidence for the negative feedback in this separation. There are not a lot of data points here because it's quite difficult to do the experiments. So if anyone has a different model system where this feedback could be measured, I think it would be fun to investigate. So yeah, to come back to where I started from, this nice movie of the deforming wave, if we now take the simple model that I presented with initial zero curvature, but still periodic boundary conditions, and we apply initial deformation, as you maybe would expect at the beginning of the wave, you see this runaway process, and you see this deformation that looks somewhat similar than as in the movie of the Drosophila. And there are a few things that are very different. So for example, here you have the active deformation on the top and the bottom here, which I think you don't see in the experiments. So there are some ideas about how to modify that to look more similar to the experiments, but I think it's a first step maybe in this direction. And with that, I want to thank Ray, who did all the experiments, who is in Cliff's lab, and then Maha worked with me on the model. Thank you.